Hello, everyone. I'm here today with Dr. Lilach Sheiner from the University of Glasgow for this quarterly ISOP member spotlight. So, Dr. Sheiner, it's great to meet you and talk over Zoom.
Great to meet you too, after we met in South Korea. And thanks for inviting me.
Yep. So, I usually like to start these off with a pretty standard question, which is what first got you interested in protostology?
Yeah, so for me, it was almost by chance. So the person who was teaching me microbiology classes, microbiology 101 during my bachelor degree, she was a parasitologist. And so a lot of the examples she gave in the microbiology course were from the parasitology world. So I got to, as at quite an early career stage, be exposed to how different and cool and parasites are and how they suit to the different environment and the different challenges that they have and all the open question and how little we know about parasites compared to everything else. And the other thing with this person, her name is Shula Michaeli. She studies RNA biology in trypanosome still.
Yeah.
And the other thing with her was that she was so enthusiastic about her science. She would talk and you can see that she can see the little microbes in her eyes as she speaks. So I kind of really wanted to work with somebody who had that level of satisfaction and inspiration in their own work. So I went to work with her for a summer project. And then I liked it so much, I stayed for a master's. And then when it was a PhD time, I consulted her, who would you go to work for if you were my career stage? And then she gave a suggestion that still directed me to another parasite, but still in the parasitology field.
Yeah. Awesome. And like I'm assuming that's when you first came across like Toxoplasma.
Yes, so her advice was, if anybody is listening to this that is at the PhD selection level, even postdoc, ask the people around you who go to a lot of meetings, who would they work for if they were you now? Because sometimes people have a good overview of what's the up and coming thing. And so she said, oh, there is this other person, which turned out to be Dominic Soldati, who just recently established genetic manipulation for Toxoplasma. So that field is going to really open up now. And that was a great advice.
So for those unfamiliar with your lab's research, can you give like a brief overview of what your lab does and what your like main like focus is?
Yes, certainly. So I kind of mentioned Toxoplasma in a passing. I study a group of parasites that is called apicomplexan. It includes Toxoplasma and Plasmodium, which are the causing agent of Toxoplasmosis, the better well known as the cat parasite that makes you into a zombie. Uh, the other one causes malaria, which is, of course, very well known, quite kind of a global killer. And for us, most of the interest in studying these parasites is to understand divergent biology. But of course, since we focus on parasite, then some of what we discover can inform drug discovery, and we do some of that as well. And feature of cell biology that I'm using the parasites to learn is mitochondria. So that's the organelle in many eukaryotic cells, almost all eukaryotic cells, that is responsible for energy and or critical metabolites like nucleotides. And so it's a very fundamental part of a cell. And so for a long, long time, mitochondria were considered quite conserved across eukaryote and across the evolution of eukaryote because of its fundamental role. And the more we delve into divergent organisms, we find that that's not the case. And I think that's where the protists are really making things really interesting.
Yeah, definitely. And I guess along the same lines, how do you sell your research to the public? Like what's kind of like that like first paragraph of all your papers about why mitochondrial reduction across APs are important?
Yeah. Also, I think I don't put my money where my mouth is because the thing that I really like about it is that we get to discover unknown cell biology by studying these parasites, because almost all we know about mitochondria is from ophistocont. It's from everything that is between yeast and human and mammals and Drosophila and C. elegans and zebrafish and all of those things. And every time we look into something that is slightly divergent than that, we discover new biology that we considered before probably ubiquitous, and it completely isn't. So this is really why I'm excited about the research. What I would typically write in my grants and when I speak to the public is I talk about the impact of the parasites and the fact that the diversion in these pathways that we didn't expect before could and is informing drug discovery. Because in effect, it does in the end. And that's the impact that people find easier to relate to. But actually, it's too bad that me and the field, we haven't found a way yet to make people understand that, in fact, it's just important to know all the biology we don't know yet,
Yeah.
even if it's not in humans.
Yeah, definitely. And despite it not being like an apistocont, Toxoplasma, Plasmodium, those would be like in some cases considered like a model organism, at least for apicomplexan. And would
Um,
you say there's any like pros and cons for working with a model organism per se?
yes, so I think specifically working with parasites, the pros are that everything is new because the field of the cell biology field had been so focused on ophistocont.
Yeah.
Almost everything we see is new. There's so lots of low-hanging fruits. So really encouraging early career researchers to consider protists for their career. But I'll speak about the cons in a minute. And then the other thing is, like we just said, that some of the discoveries really have a chance to inform drug discovery or drug
Mhm.
development or regimes of drug usage and diagnostics and surveillance. So the impact could be beyond the discovery side of the biology. Um, yeah, the cons are that the bigger fields are still not used to looking at something new that comes from the parasite and say, oh, wow, here is a new biology, but more, oh, wow, here is this obscure parasite biology that we don't care about because it's not going to teach us about what we care about. But I think
Mhm.
that opinion is changing. And there are many examples where something has been discovered in the parasite because the parasite does it in an extreme way and then was found also in human or in mammals, like RNA editing, for example.
Mhm.
Um, so I think that notion is changing, but it's still a bit of you kind of got to convince people, scientists and journals to take these discoveries as a big discovery because it's new biology.
Yeah.
And the other cons is that parasites are harder to work with. They replicate a little slower. They're smaller. They have to grow inside the host, so there are different contaminations. You got to get permissions to work with them because they are pathogenic.
Yeah.
So there are a few more technical barriers.
So the ones you work with in the lab, are they like pathogenic? Like, are they like knocked out at all in order to like lower the pathogenesis, or like how is that kind of handled?
So the toxoplasma we work with in the lab, we work with the tachyzoid stage. So this is a stage that causes brain damage when you have like acute toxoplasmosis.
Hmm.
But at the same time, this is the state that you cannot get through aerosol or through eating them. You have to inject them to yourself.
Oh
Um,
wow.
so they are not really pathogenic because you're not injecting them to yourself. And we try we don't work with needles and take all the right risk mitigations. We some people in the field are working with the cyst form, and those are a little more infectious.
Okay.
Um, and toxoplasma as a whole is only pathogenic when you become immunocompromised. But at the same time, it also stays for life. So it's not immediately pathogenic, but it could become pathogenic.
Okay. You've developed some really cool tools for working with Toxoplasma. Would you be able to like talk about one of these and like what it has led to?
I think the one that I will probably bring up is that, and this is not just my lab, it's a big collaborative effort. So our greater team has developed and established structure, like the collaboration between structural biology and genetic manipulation and cell biology, and the pipeline that connects those two forward and backward in toxoplasma, and specifically for mitochondrial complexes, so complexes of the respiratory chain and ATP synthase and the mitochondrial ribosome.
Yeah.
And so I think in that sense, that's a sort of a tool or a pipeline that we developed that wasn't available before. And our greater team has probably published the most structures, certainly for mitochondria, that are then also backed up with, so the observation from the structure are backed up with genetic and cell biology validation. And so we really get a proper mechanistic understanding of things from the structure function side.
Awesome. Now back to ISOP and prosology in general, I guess. You're a relatively recent member of ISOP. Do you have any initial impressions of the organization and the like broader protostology community at all?
Yeah, I think what I really like about ISOP, and this is also why I became a member, is that, unlike what I was saying about the general cell biology community, this is a community that cares about divergent biology and that appreciates new biology from wherever it came, regardless of, whether that's going to help us tomorrow to cure cancer or diabetes. So that's a lot of fun. And I think because of that also, there is much more effort to be very broad in the topics, because the parasites live in, or protists live in
Mhm.
different niches. And so the topics of their biology that come up are completely different. So there is a big variety of biological topics that we're looking at, and as well as a big variety of organisms. So I really like that.
Awesome. And then like the organization, did you like you were in Korea for the last ISOP conference. What are your impressions of that?
Well, what I really liked about it is that there was a call at the beginning to the community to propose sessions, and that call was also accompanied with some funding that came to bring in some of the speakers, and it was accompanied with a focus on making sure that if you put together a session, there are people at different career stage, and there is a divergent group of speakers, so that there is a good representation of also the diversity of the researchers themselves in the field. So I really liked that we had that opportunity as a community to bring that forward. And then I also really enjoyed the resulting sessions that were presented in the conference. Also, I really liked the venue. I mean, South Korea
Yeah,
was.
it was great. You mentioned your previous mentors from undergrad and your PhD. Is there anyone else that really influenced your scientific career?
I think so many people, it almost feels unkind to only speak about a few. I think the cool thing about us, the scientific community, scientists, is that everybody is keen to help their peers. So if you go and ask people, it's very rare for somebody to say, "Oh, I'm not going to give you an advice because whatever."
Mhm.
Hardly ever happens, right? So there are people starting from like my teacher in my high school who made me really excited about biology, and the person I spoke about in the bachelor degree, and then Dominique, who I worked with as a postdoc, and both Shula and Dominique are women who are very successful in science, who are very impactful. So that was great to see.
Yeah,
Just as an example, that kind of gives you some sense of empowerment. And then my postdoc supervisor, Boris Trippen, who was so kind in letting me play around with mitochondria, even he didn't think that was very interesting. So I could build my own research persona, and he was very generous about that. And then throughout my career from postdoc until now, there was always somebody to ask for advice in some form or shape, about my career or about my science or about my um, personal life as a person with a very demanding career, all of those things. So
Yeah.
yeah, it's like, a series of mentors and people who helped.
Yeah. So mentorship is very important. Is there any other advice you have for early career researchers?
I think just communicating with people around much, much more than people feel comfortable doing. Firstly, because it's easier. So if you speak to somebody, it's a much more, usually a faster and a more efficient way to get information than sending an email or reading a paper
Yeah.
or even watching a video like this, because you just get your answer very fast. And I
Hmm.
also think it's more memorable when that depends on how people think, but for many people, it's more memorable when somebody tells you something and gives you a personal example. So go and speak to people with, whatever question you have. And I think a side note of that is going to a lot of meetings. And when you go to meetings, be very active, get people to listen to what you have to say, either drag them to your poster or ask them after your talk, what did they like about it? Do they have any advice? Do they have any comment? Also put yourself out there for criticism. I think that really helps. And again, just this communication, it's so efficient. And then also you have a network and it's very helpful later on.
Definitely. Um, the last question for this, do you have any current or future projects within your lab that you're particularly super excited about
Oh,
that you're
yeah.
willing to share?
You wrote me that question in the prep for this interview, and I'm like, you're asking me to choose between my kids. Who is my favorite kid? I can't say that. Um, I can say that the ones that are the best funded at the moment, but that's sometimes random. What grant or what paper we published recently or got recently.
Yeah.
Um, so one is about the mitochondrial ribosome, which we recently shown he's made of more than 50 pieces of ribosomal RNA, which is very, very unusual for ribosomes. And we're now trying to understand how does this assemble. So we published the structure so we know what the final product looks like, but we're trying
Mhm.
to understand how does this assemble and how it is regulated in response to what the parasite experiences. And the other thing is also when we studied the respiratory chain and ATP synthase, we discovered a lot of parasite specific, in fact, not parasite specific, but mysozoan specific, so
Oh,
protist specific components of the chain, of different complexes of the chain. And we saw that though many of them are responsible to mediate interaction between complexes and other complexes. Um, and in fact, that's what I was talking about at the meeting. And now we're trying to understand why this is important in the various conditions that the parasite have to survive. So how do they contribute to the parasite's ability to infect, to survive in the animal, to disseminate, now that we understood what they're doing to the parasite in culture. So I guess
awesome.
those are two of them, yeah.
Yeah.
But many aspects of mitochondrial research in the lab that I'm super excited about, and I just don't have the time to speak about all of them anymore.
Yeah. I excited to like see that like future work. So, for people that want to learn more about your lab or to contact you, is there any like specific site or anything you want to lead them to?
Yes, so I try to be on quite a few social media. So if you search for Shiner Lab, you will probably find our X account, our Blue Sky account, and LinkedIn account. I have to admit that LinkedIn is the one that I check the least,
Yeah.
but I still check. So give me a couple of weeks and you will see that I come back to it. Um, and if you look for Glasgow University, Lilach Sheiner, you will find my staff page, and there we'll have links and emails and everything else. And please do that because I love speaking to people about science or career or whatever.
Awesome. Well, I really appreciate your time, especially on a Friday afternoon for you. So, th it, thank you a
10 o'clock?
lot.
Yes.